Hi, and welcome to this video where we're going to be going through this AS level biology, depth in biology paper from 2016. So the first thing you need to do, if you haven't yet done this paper, is to follow the link underneath and get the paper in front of you. Print it out and do the paper. Alternatively, if you're just finding this video and you're not one of my class, what you could do is you could sort of listen to the introductory explanation of each question, and then you can pause the video at the appropriate point, write down your answers for the question on screen, and then see how you do in the Marc scheme and sort of do it live like that. Okay, so let's get going. We'll start with question 1. Okay, so the first question down here is about breathing, the mechanism of breathing, okay, inspiration and expiration. So here we have two diagrams, and it says, which of the two represents the body immediately after expiration, which is exhaling? Let's zoom in here. Okay, so which of the two represents the body immediately after expiration? Okay, so it's A, basically. How do I get to that? Okay, so A, the reason is, if we look here, the diaphragm is relaxed and flattened. And second of all, we can see that here the rib cage is sort of up and out, whereas here the rib cage has come down. Okay, so we need to talk about diaphragm, relaxed, sort of, oh no, sorry, I made a mistake there. So the diaphragm is relaxed and sort of curved up. Actually, this is contract. When it contracts, it pulls down. So relaxed and curved up. And the second thing is rib cage is sort of down. Okay, so Marc scheme. Here it is. Oops, that's one wrong. Sorry, this one. Here we go. So here are the different things you could have said. The lungs are deflated or less inflated. The diaphragm is domed or curved up or arched or not flat or is relaxed. Notice it says ignore pushed up here, because pushed implies an active process, and this is relaxed. Also, the rib cage is down or lowered. Talking about intercostal muscles is not awarded. There's not any marks given because you can't really see the position of the external intercostal muscles in this diagram. The muscles are not there. Now, why can expiration be a passive process? There's two reasons. First of all, if you inhale and then you let your ribs fall, gravity can cause your ribs to fall. And second of all, there is kind of an elastic recoil up with the diaphragm. So you have to actively contract to pull it down, and then it kind of pulls up elastically. So let's just go straight to the Marc scheme. And here it is. So here's the Marc scheme for that question. It does not use muscle contraction. Muscles just relax. So rib cage falls due to gravity, or lungs elastic, so we will recoil. So yeah, those are the things that we said. You need to be specific though. Always look at what it says to ignore, because you want to check that you didn't say that. Often students won't get marks because they won't be quite specific enough, like this. Rib cage moves down, as this could involve muscle contraction. It doesn't necessarily say that it's passive. Okay. Moving on to question 3. Some chemicals can act as allergens. If these allergens are inhaled, they can cause breathing problems. Allergens can cause the smooth muscle on the walls of the airways to contract. Suggest the effects this muscle contraction has on ventilation. Okay, so what do we need to talk about here? Well, we need to talk about the bronchi and bronchioles. Whoops, can't spell. Bronchi plus bronchioles. These are the things that contain the smooth muscle. And the smooth muscle will take a sort of wide lumen and constrict it down to a narrower lumen. So we need to talk about the lumen width and the fact that it's going to get narrower. And then we probably want to talk about the resistance to airflow. So with a smaller diameter, with a smaller lumen, we're going to have a higher resistance to airflow, which is going to make ventilation more difficult. Okay? Let's see how the Marc scheme deals with this question. Here it is. So there is a reduction in the lumen diameter of the bronchi or bronchioles. And you've got to talk about those, either one of those words. If because of this slash here, that means you could use either the word bronchi or bronchioles, but you must use one of those two. They are underlined terms, which means that they are essential for the mark. So therefore it's harder to exhale, or there's more resistance to exhalation or more resistance to ventilation, so less air can be exhaled. So more air remains in the lungs, and it's also harder to inhale, so it's harder to ventilate. So more resistance to ventilation, increased breathing rate or gasping. Yeah. So again, let's look at the ignore, because we need to make sure that we're kind of really speaking scientifically. Ignore just it's hard to breathe or struggles to get breath or any of these things here. Okay, they're not good enough. Okay, there is more resistance to ventilation or more resistance to breathing. I think that would that. You know, actually, I don't know if you get the mark if you just said there is a reduction in the diameter of the lumen and therefore there is more resistance to breathing. You might have to say there is more resistance to breathing in and out. Okay, so in for inhalation and out for exhalation. Yeah. Okay, moving on to the next question. Question B. So here we have a spirometer trace, so we can see all the different components of lung ventilation or lung volume, sorry. Okay, so what's what? Okay, residual volume, total lung capacity, tidal volume, and vital capacity is what we're looking for. If you're doing this sort of live, just quickly pause and see what you think. Okay, so I'm going to label it on the diagram. So total lung capacity is D. And C is what's called the vital capacity. Tidal volume is F, which is kind of just what you breathe in and out sort of normally when you're not exercising or actively breathing in a lot and breathing out a lot. It's just your normal kind of standard breath volume in and out. So what have we got? Let's just see. Tidal volume is F. Vital capacity is C. Residual volume, okay, that's H. So that is the volume of air that's in the lungs that you can't really get rid of. Even if you exhale to your absolute maximum, there's still some air in your lungs that you can't push out sort of forcefully. And D is total lung capacity, so we've done everything we need to there. Okay, on to 2. Volume C can be measured using an instrument such as a spirometer. What breathing instructions would be given to a person whose volume C was being measured? All right, let's just go back up here because I can't remember which one C is. Okay, vital capacity. So vital capacity is from a maximum inhale to a maximum exhale. So the instructions will be something along the lines of breathe in until your lungs are completely full of air and you can't get any more air in them. Then, put your mouth on the tube and then breathe out to your absolute maximum and you can't push any more air out of your lungs. This volume change would equal your vital capacity. Let's see what the Marc Scheme says about those 2 questions. Okay, so yeah, breathe in as deeply as possible and then force as much air out as possible. Okay. Interesting. Always look at the ignore or the do not credit, because I think, these are the things that we can easily mistake if we don't speak, correctly. Do not credit all the air pushed out of lungs. Why not? Well, because you can't push it all out of the lungs because of that residual volume that will stay there. Okay, just the maximum possible, not all. Moving on to question 2. Ooh, look at this interesting chart. Okay, immediately you might see this, you might think, ooh, this looks horrendous, but just breathe, take a second, and you're going to figure it out in a moment. So let's first of all just look at the charts. Actually, I think that's a good idea before you tackle a question. What's going on? Just sort of follow it around. Okay, well, we've got fertilization, then we've got an embryo, and then we have a diploid plant, and then we go to some spore mother cells, and then we have some spores, and then we have a haploid plant here, a gametophyte plant, which kind of makes its female and male gametes, and then we go around. So we're talking about a life cycle. Here's another life cycle of a different an adult. So this is not a plant. This looks more like a, yeah, this is a human because we've got sperm and eggs. So we've got an adult. That's weird, isn't it? A haploid adult. So maybe this is not a human. And then we're going to go to sperm and eggs, and we fertilize to get a diploid zygote. Okay. Place a tick in each row of the table to indicate the type of nuclear division that occurs at A to E. Okay, so what's going on at each step? Well, let's look at A. So A, well, it looks like we're doing meiosis there because this is diploid and this is haploid, so we're going from diploid to haploid, so I think that's meiosis. B, we're going haploid and these are still gametes, so these are still haploid, so that's mitosis. And C, this is going to be diploid and this is diploid, so this is mitosis. All right, moving on to this next little bit. What's going on at D? Well, we're going from a haploid adult to haploid sperm and eggs. So that, weirdly, I think that's mitosis. Even though we're producing sperm and eggs, I think it's mitosis. And here, this is going from diploid to haploid. So that is meiosis, okay? So this is a real question that's going to kind of make you think and not just recall information. Because if you just recall information, you might go, oh yeah, D, sperm and egg, that's meiosis. But actually, no, because it's from haploid to haploid. Okay, so that's a tough one. I'm just going to check and see if we got this right. Tick, 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 tick. Okay, is that what I said? Dun, dun, dun. Yes, it is. Fantastic. Okay, moving on. Here is a life cycle diagram. Different phases of the cell cycle. X, Y, and Z represent checkpoints in the control of cell cycle. State all the letters that represent the phases of interphase. Well, there's loads, okay, because M is mitosis, and I think C is cytokinesis, as far as I can tell. So this is the actual splitting of the cell here. So what have we got? So we've got G1, S, and G2. Suggest what is being checked. So interphase is all, let's shade it in, all of this, oops, that's not very good, but all of that is interphase, all that stuff around there is interphase. I've got to colour this in nicely now. I really like colour, you may notice if you've seen my videos. Ta-da! Okay, there's the interphase part. Okay, suggest what is being checked at checkpoint Y on figure 2.3. Okay, so where's Y? What's going on? So, okay, so this is after S phase. So I would say at checkpoint Y has the DNA been replicated correctly? Has all the DNA been copied correctly? Okay, let's check that. Let's check 1 and 2. G1, S, and G2. Idea that checking that DNA is replicated correctly. Yeah, good. So for this, just in case you wrote G0 here, if you wrote G1S and G2 and G0, I think you still get the credit, okay? Because that's an ignore. So you could have written these things, but basically you don't get marks off or you don't get marks for those, okay? You ignore, pretend they're not even there. However, if you included M or C, that is not correct because M is standing for mitosis and C is for cytokinesis. All right, on we go. Okay, table 2.1 indicates the relative time spent in different phases of the cell cycle for different types of cell P, Q, and R. Which of the cells takes the shortest time to divide? So I think this almost seems a bit too easy. I think we just add them up. So 18 plus 50 is 68, plus 13. Who am I kidding? I need to do this on my calculator. So you just tally them up and write them down to the side. So as my calculator, 18 plus 50 plus 13 plus 19 equals, okay, so we've got 100 over here. Are they all going to be 100? I bet they are. 18 plus 25 plus 11 plus 16 equals 70. Not 100. And this is 100. Okay, so it's Q. Q is that one. Suggest why cells P spends twice as much time in phases S than cell Q. Okay, so this one, 50, that is 25. Okay, so, well, it may just have, cell P may just have twice as much DNA. Okay? Because S phase, what's going on there, S phase is the copying of the DNA synthesis phase. So cell P has twice the amount of DNA as Q. What can be deduced about the behavior of cell R? Give reasons for your answer. Well, cell R is very interesting, isn't it? So cell R, it is spending 100% of its time in G1 or G0 and 0% of its time in any other phase. It's not copying its DNA. It's not G2. It's not getting bigger, ready for dividing, and it's not dividing. Okay, so what is it doing? It is a quiescent cell. That means kind of quiet or sitting out. So it's in G0. So it's in the G0 phase. G0, not actively dividing. We can use the word quiescent state. Could be something like a nerve cell. Potentially, neurons. Your brain cells don't divide really. Once you've got them, you've got them. So treat them nicely because you don't want to damage them and get rid of them because they won't come back. Okay, let's go to Marc Scheme. So Q, it needs to synthesize, has more DNA. That's what we said. Or any valid point. What are the valid points they say? So reference to P being from an organism at a lower temperature. Yeah, that's a valid point. So if P is an organism that lives in, I don't know, an Arctic sea in midwinter, under the sea ice where it's very close to freezing, chemical reactions, including the ones catalyzed by enzymes like making DNA, are going to take a long time. Down here, so this is the cell which is in its quiescent state, so it spends all of its time, does not leave G1, or it does not leave G0, so it's not dividing or replicating. It's specialized or it's differentiated at any valid point, so reference to having reached the end of its development, for example, a neuron. There we go, there's an example, that's what we used, neuron. Or an erythrocyte, for example, a red blood cell, B memory cell, etc. Or potentially it's dormant, yeah, all that sort of stuff. These ignore here, these are kind of going down the wrong avenue, really. If it was a stem cell or a cancer cell, you'd expect it to be dividing quite rapidly. Okay, moving on. All right, an experiment was carried out where a student observed cells in different tissues under the microscope. The cells were undergoing mitosis. 200 cells were observed for each tissue. The number of cells in each stage of mitosis was recorded. Okay, so we've got V and W, and we can look at the different phases of mitosis and then work out the total. The student had expected the results observed for tissue type W would not be significantly from tissue type V. Identify the pieces of evidence in table 2.2 that cause the student to suspect the results might be significantly different. So we're basically just kind of spotting the difference here. What is significantly different? And I would say, you know, this here, prophase, W looks like it's got more time in prophase. These are pretty close. These are pretty close. And then this is also different. Okay, so we're talking about the fact that V appears to be spending more of its time in telophase, whereas W is spending more of its time in prophase. And that's going to just kind of provoke interest and thinking about, thinking at what is, you know, is this really different? Is it a significant difference? Okay, so part 2, so D part 2. The student decided to analyze data using a statistical test. And they said, do the t-test. But why is this not a good idea? Well, let's think about this. How do you do a t-test? Well, you need the mean and the variance Do you see a mean and a variance here, or a mean and a standard deviation? No. So there's no mean, there's no standard deviation. The data is not normally distributed as well, you could say. So all of those things would be worthy of the mark. Let's just quickly look at the Marc scheme and check, whoops, that we got that right. Okay, so yeah. So this was for the first part. W has more cells in prophase and fewer in telophase. And that second part, t-test is used to compare means. And also we can't calculate standard deviation from this data. Okay. Ignore reference to average instead of mean. You've really got to talk about the mean and not the average, because remember there's 3 different types of average. You've got the mean, the median, and the mode, and t-test is about comparing means. Okay, on to the next one. I feel like we're going to do a calculation here. Yes, we are. Okay, so chi-squared. The chi-squared test can be used to analyze the data. Now, I'm not sure if you've done chi-squared my year 12s. We'll see how you did anyway. I'll briefly explain it. It is some statistics that we will learn in year 13. It's in your year 13 textbook. That's why we haven't covered it, although it could be assessed if you were doing AS, but don't worry, we will cover this in the future. Okay, so this formula, let's break it down for you, for my students who haven't looked at this. This is about comparing what you observe with what you expected. So for example, in this case, the prophase, you expected 65, but you observed 85. So there's a difference of 20. So O minus the E is the difference, observed minus expected. And then you have to square it. You have to square that difference. So 20 squared is 400. And then you have to divide that squared value by the original expected value. So that's 400 divided by 65 is 6.154. And then hopefully you recognize this symbol here. What you do is you have to sum all of those. Okay, so if you fill out the table, you might have done it correctly. So let's do that. Okay, so first of all, we need to get our observed and expected values. So the expected values are tissue type V, and the observed is W, because what we expect was that everything would be the same as V, and then we look at W. So let's look at, first of all, the observed, that's W. We're going 85, 59, 6, 50. So 85, 59, 6, and 50. And then the expected is 65, so it's 55, 7, 73. 55, 7, 73. 55, 7, 73. Okay, so now let's do the O minus the E. In this case, it's 4. And then if you square that, that would be 16. Here, 50 minus 73, that's 23. In fact, it's going to be minus 23, but we square that to get rid of the minus sign. So 23 times 23 is 529. Now, in this last column, we have to divide by the expected. Now, it's worth, for me, highlighting this here, because we need to divide by the expected values, not the observed values. It is sometimes easy to make a mistake and divide by your wrong number. You could have divided by observed. I've done that in the past myself. Okay, so we want to do 16 divided by 55. The expected, which is 0.29. I'm going to keep 3 decimal places because that's what they've done. 0.290 is actually 291 actually if I round it up. And this one, it's 529 divided by 73. 529 divided by 73 equals, that's 7.247. If I'm doing 3 decimal places. And now I'm going to total up this whole last column for the totals down here. So I've got my last number in my calculator already, plus 0.143 plus 0.291 plus 6.154 equals 13.83. To 2 decimal places. Let's check that that is correct. Yes. Oh, I lose a mark. Okay, I went to 2 decimal places. Silly. I should have kept it to 3. answer should be 3 decimal places to be consistent with the rest of the table. I was doing that with my working, but then I got slack. Very silly. So I should keep that as 3 decimal places. So there's a moral of the story for you, and I have to round that correctly. It's 835. So be consistent with decimal places. If you didn't do that, just make a little note to yourself. Keep DP consistent throughout. Okay, all right. The value of chi-squared can be used to conclude whether results for cells and tissue type are significantly different from those with V. It's very similar to t-test. We're talking about looking at this calculated value and saying if it's bigger than the critical value. Again, like the t-test, we have to do something called degrees of freedom, and it tells you how you work it out. It's the number of categories minus 1. So how many categories are there here? Where are the categories? Well, they're up here. The categories that you can have are prophase, metaphase, anaphase, and telophase. That's 4. So there's 4 categories, minus 1. Degrees of freedom is going to be 3. Here's our degrees of freedom table. So remember, we want to look at here 3, and it says here. So we want to basically always use. We're generally always using this 95% confidence, or is it the 5% confidence? No, it's the 5. Yeah, it's that one. So yeah, sometimes I get confused between which one we're using, because this means that there is only a 0.05% chance that this level of difference is down to chance. So here it's 7.82. So is our calculated value larger than that? Yes, it is, 13.835. So we can say chi-squared is 13.835, which is larger than P 0.05 value of 7.82 with 3 degrees of freedom. This means that the difference is significant and is not due to chance. If you wanted to go further, you could actually say that because the calculated value is bigger than that, we can actually say that there is only 0.01% probability, or there's a probability of 0.01, I should say. There's only a probability of 0.01 that that this is just due to random chance. Okay, so it is actually somewhere between. Our calculated value is somewhere in the middle of these 2 values. Okay, so let's look at the Marc scheme there. So first of all, you get marks for filling out this table. We've already looked at that. 3 degrees of freedom, and then in terms of the conclusion, so the calculated value, this is the one we're looking at, this row here, that one there. So the calculated value is greater than the critical value at P 0.5. So the difference is significant or is not due to chance. And then we can even go, this is the 4th point here, that the chance that the difference is due to chance is even less than 0.01. Okay, so something I forgot to say, silly. Hopefully I still got full marks for that. Is that we therefore reject the null hypothesis. Reject the null hypothesis. I've got to say that. Okay. Okay. Moving on to the next question. Okay, question 3. Polymers are important molecules that have structural and functional roles in organisms. Chitin is a polymer that's found in insects. It's a macromolecule similar to a polysaccharide, but it's sort of polymers of this N-acetylglucosamine, and we see the monomer below. So how is it different from a composition of a monosaccharide sugar? Well, there should be hopefully one key point that you managed to identify, and it's right here, okay? There, it's an atom. It's the atom nitrogen, which is not present in a sugar. So, the second thing, which monosaccharide sugar does glucosamine most closely resemble? Well, look at this. This is interesting. How is that worth 2 marks? How is it worth 2 marks when it seems to be a one-word answer? Well, the answer is it's not one word. It's glucose, but what type of glucose is it? This one is beta glucose because the OH is facing up, so it's similar to beta glucose. Okay. All right, on we go. Using your knowledge of the formation of structural polysaccharides, describe the formation of the chitin molecule from its monomer and predict its structure. Okay, so this is basically a question that's asking you about your knowledge of cellulose, but asking you to apply it to a novel situation. Okay, so we're not talking about beta glucose, we're talking about N-acetyl glucosamine, and we're talking about 1,4-glycosidic bonds. Okay, so right, it's the same stuff as cellulose. So we've got N-acetyl glucosamine. We've got we need to talk about the fact that each monomer will be flipped 180 degrees with respect to the previous monomer in the chain. We've got to talk about a straight chain. We've got to talk about hydrogen bonds between chains. We've got to talk about maybe sort of microfibrils building up into macrofibrils of this. And we've got to say, you know, therefore it's very tough, gives strength, support. And it already told us about 1,4-glycosidic bonds. So yeah, we can, you know, we maybe can sort of say that again. Beta 1,4-glycosidic bonds, I guess. Beta 1,4 is what we would say. Beta 1,4-glycosidic. Okay, so those are all the things that we want to talk about when we write this answer. Let's look at the Marc scheme now. Okay, here we go. So first of all, there's the other 2 questions that we didn't show the Marc scheme for. So nitrogen, beta, glucose. All right. There's some stuff that I didn't say. So what did I actually say? What marks did we get? We talked about alternate monomers, upside down. We formed a straight, linear, unbranched chain, similar to cellulose. There's some things that I didn't say, which I should have done. So I probably only got 3 marks out of 4 for that. I didn't actually say that in chitin glycosidic bonds we form by condensation. Molecules of water produced or released because of the position of the OH on carbon 1. So yeah, those are the 6 things you could have said in terms of how it's formed and the structure. Now, where did I go wrong on that? I didn't. Classic mistake from me, rushing. Should know better. So what I did there is I didn't really analyze the question enough. This is why you got a highlight. Okay, so what I mainly talked about this, didn't I? I mainly talked about its structure. I didn't describe the formation of the chitin molecule because I was rushing. Okay, so describe the formation of the chitin molecule. I was talking about condensation reactions between the 2 N-acetyl glucosamine molecules, forming water and forming a beta 1,4-glycosidic bond. Okay, so you've got to do both parts for the 4 marks. Okay, moving on. All right, here we've got a photomicrograph of the trachea of the honeybee. The trachea of this honeybee is infected with honeybee tracheal mites. How awful. Acarapis woodii. Some of these mites are labelled M on figure 3.2. The trachea and tracheoles of insects have circular bands of chitin. One of these bands is labelled C on figure 3.2. Well, remember these bands, they prevent the trachea and tracheoles from collapsing. They keep them they give them support and prevent them from collapsing down. It's a bit like the bands on a straw in that bendy bit, which kind of prevent it from collapsing in that point. The mites use their mouth parts to bite through the walls of the trachea, then they feed off the hemolymph, which is a blood-like liquid that bathes the cells of the honeybee. Suggest one way in which the presence of the mites might affect the honeybee. Well, it's going to prevent the movement of air or the diffusion of gases down the tracheoles. So remember, the air can be sort of actively moved into and out of the tracheoles in some insects, for example, locusts, but it's also just a route via which gases can diffuse down those tubes. So it's going to prevent either the movement of air in and out of the tracheoles, and it's definitely going to slow down the rate of diffusion of oxygen and CO2, for example. Let's look at the Marc scheme. So the rings of chitin, they support and they prevent the trachea from collapsing, which is what we said. And then here, lots of things we could say. It restricts the airflow, deprives the tissues of oxygen, which is what we said. Also, this is some stuff that we could have also said, good points here. The mites themselves might use up some oxygen. Also, they could act as a vector for disease transmission. Bees can get viruses, by the way, so maybe these mites can carry viruses to bees, or the mites might release toxins. Accept reference to inflammation or immune response. Great. Okay, moving on. Moving on to question 4. At this point, if you want to take a break, I suggest this would be a good point. I've got a few more questions to do, so maybe pause, grab a cup of tea, grab a snack, come back. I'm going to work through the rest of the questions. Okay, welcome back if you did take a break. So now we're on question 4. So here we have a fungus, which is called Amanita citrana. Placed in the egg or petri dish. The petri dish contains starch. We've got the thread-like hyphae growing out there, a few centimeters. Then we use iodine solution to test for the presence of agar. So, to which genus does this fungus belong? Well, this is not something you're expected to know. You just have to remember that the last two, well, the binomial name contains the genus and then the species. So therefore, the genus here is Amanita. And the species is Citrina. The region of yellow staining, so that's this bit, okay, in figure 5.4 includes part of the agar where the fungus had not grown yet. What does this pattern indicate about the action of the fungal enzymes? All right, okay, so what's going on here? Well, the fungi release enzymes extracellularly. Hopefully you remember that. So we can see that from this picture because where the hyphae have extended, they've released enzymes, but those enzyme molecules, those proteins, can diffuse through the agar further than where the fungal hyphae have actually got themselves. So that's what this is showing. So basically, we want the word extracellular enzymes. We could also maybe talk about saprotrophic nutrition, because that kind of describes how they feed, like so. Okay, let's check the Marc scheme for that also. Let's just check that's what they've said. So Amanita, starch digestion in the region where the fungus is not present, enzymes are released, they're extracellular or they're secreted, okay? All right, back to the paper. Okay, on to B. Right, we've got quite a extensive experimental method here. So when we reach one of these in a paper, we whip out the highlighters and we really try and focus on the key ideas. Okay, so 5 centimeters of an alkaline solution of lipid. Water bath, 20 degrees C, that's important, and left to equilibrate, that's right. A few drops of an indicator were added to the wells of a white spotting pile. Okay, and this tells us, I'm just going to do that, the indicator is pink above pH values of 8, pH 3, and colourless at pH values below pH 3. Okay, do that pink because it reminds me of the indicator. Once the lipid solution had equilibrated, 1 centimeter cubed of 0.5% lipase at the same temperature was then added to the test tube. For 5 minutes at 30-second intervals, the solution was stirred and a few drops removed from the test tube and placed in a well on the white spotting tile. The time was recorded when the solution and indicator did not remain pink. So basically this tells us that the pH has dropped below 8.3. Okay, I'm putting that in pink because it's about this indicator. You've repeated 4 times at 20 degrees C and then a further 6 temperatures. Okay, so time when solution did not remain pink. So why is pH not a controlled variable in investigation? Well, because pH is used, it's not something you want to keep constant. This is pH is how it's the dependent variable really. It's how we're measuring It's how we're measuring, because it's how we are measuring the digestion of the lipid into glycerol and fatty acids. That's the reason. Okay, so we want to see those fatty acids being produced, drops the pH below 8.3, and then we can detect that. In the Marc scheme, that's what it says. pH is the dependent variable being measured because fatty acids are produced. Okay, pH indicates the rate of the reaction. Yeah, it is sort of the dependent variable. If I was being very picky, I wouldn't say it's the dependent variable. The dependent variable is the time until the solution did not remain pink, but pH is kind of involved in that. Okay, identify one variable that has been controlled in this procedure. Well, this is where that highlighting comes in. Look at all these things that were controlled. Well, we've got 5 centimeters of alkaline lipid. You could have said that. Few drops, that's not very controlled. This is nice and controlled here. 1 centimeter, 0.5 lipase, so the amount of lipase, and the percentage lipase solution, and yep, that's pretty good. So those things, okay. So I will say 5 centimeters of lipid. Or, well, the volume of 5 centimeters of lipid solution. For example, identify one variable other than pH that has not been controlled in this procedure. Again, we go back up here, we have a little think about it. And here's something that maybe you might want to talk about. The number of drops of the indicator, that wasn't very controlled. Yeah, I would probably say that, the number of drops of indicator. Bet there's loads which I haven't thought about. Let's have a look at the Marc scheme. Okay, so these are all the things that were controlled. The volume of the alkaline lipid solution. Notice, again, Marc scheme, very important. The underlined words. If you don't put these words, you don't get it. So the volume of the lipid solution. Concentration of the solution. They love the word solution, don't they? Concentration of the lipase enzyme solution. So always say solution. Volume of the solution. The temperature, time intervals between testing, the 30-second intervals. Now, what do they not control? Here are the things they did not control. The concentration of the lipid solution, that's a good point. The volume of the indicator, because they said a few drops. The number of drops of indicator, that's what I said. The volume of the sample mixture removed, that's true. They didn't specify that. And the number of drops of sample mixture removed. All those things were not controlled. Okay, the procedure required the solution to be stirred and then drops of the solution to be placed on a white spotting tile. Suggests why this procedure was followed rather than simply adding indicator to the test tube, stirring solution and looking for color change in the test tube. Well, I guess there's a possibility of the indicator affecting the enzyme action. Isn't there? And we don't really want that to happen because that introduces other variables. So that's what I would say. We'll look at the Marc scheme for that in a second. What can be concluded from the results in table 4.1 about the optimum temperature for lipase activity? Okay, well, let's go up. Which one is the optimum? Well, we're looking for a very fast time for it to turn pink. So that's quite slow. That's a bit faster. This is pretty fast. This is pretty fast. And all of these, well, yeah, it's probably 35, isn't it? It's probably around closer to 35 than 30. Because, you know, most of them are the same for 30 and 35. However, this one, it's a little bit faster. Maybe that's an anomaly, but it's between 30 and 35, certainly. Probably a bit closer to 35. It's only a 1 mark, so yeah, between those two. Let's have a look at the Marc scheme now. So 4B, well, the white spotting tile, this is something that I didn't think of. This is a good one. Looking at the small volume against the white background makes it more easier to see the color change. And what it said here is a nice explanation of that. It provides a contrasting background color with which to look at the pinkness. This is what I said. The indicator might affect the progress of the enzyme reaction, potentially. And this is another thing you could have said. Better temperature control as test tube not taken in and out of the water bath constantly to look at it. Yeah, that's also a very good point. Yeah, the optimum temperature is between 30 and 35. Oh, look at that. Sometimes we've got to have this degrees C must be stated once. They're very picky on this, okay? So don't just say the optimum temperature is between 30 and 35. Got to use the units between 30 degrees C and 35 degrees C. Ignore around 35. Yeah. Okay, back to this question, this next question here. Describe the two different ways in which the procedure could be modified to obtain a more accurate value for the optimum temperature of lipase activity. Okay, right. The major thing is that we need to we need to interrogate this area here more in more detail, don't we? Because we don't really know where the optimum temperature is in this range. It's somewhere in there, but we don't know where. So what we would suggest is we, repeat the experiment and we do 30, 31, 32, 33, 34, 35. We look at a greater, not a greater range, we reduce the intervals of temperature. That's what we've done there, okay? So now the interval of temperature here is 5 degrees. The interval of temperature here is 1 degrees. So if we do that and repeated the experiment using 1 degree temperature changes between 30 and 35, then we could more accurately identify and say, oh look, the optimum temperature is exactly, let's say, 33, not 32, not 34, but 33. Okay, so that's one thing we could do. Now let's look at the Marc scheme because I can't think of the second one right now. Okay, so this is what we said. Use more intermediate temperature values, for example, at 1C intervals within that range. Okay, so in the 30C to 35 degrees C range, so there's 1 mark there and 1 mark there. That's also true. Yeah. So to look at the other, to try and figure out what else you should do. Once you've had that first idea and you're not sure about the second thing, you should go back and read the method in depth. So let's just do that for a second. If we go back and look at that method, we sort of focus on this part here. Okay, for 5 minutes at 30-second intervals, the solution was stirred and a few drops were removed from the test tube and placed in a well on the spotting tile. And the time was recorded when the solution did not remain pink. So this is a little bit subjective, isn't it? So because it's subjective, we could use a colorimeter to improve. And this 30-second intervals, maybe 10-second intervals or something. It's going to be quite difficult to do it sort of regularly, but that would give us a more accurate reading here, wouldn't it? Okay, so back to the Marc scheme. So take samples at more frequent intervals, like more frequently than every 30 seconds. For example, every 15. Look what they said here. Time interval must be experimentally workable, so should be from 10 and less than 30. So if you said every 5 seconds, that's like impossible, isn't it? Because you can be like take, drop, take, drop. It's just going to be too tricky. Use of the colorimeter. Color change would be less subjective or biased. Or we could just do away with the whole indicator thing entirely and use a pH meter to obtain a numerical value for the pH, and then we could accurately measure when the pH drops below 8.3 with a bit more precision. Okay. All right. On we go. C. Ooh, look at this. Our first essay question. So we're going to take a bit of time here to kind of unpick this. There are 2 models for the mechanism of enzyme action. Outline how changes in temperature can affect these mechanisms of lipase action. Okay. All right, so there's loads of stuff we can say here. Okay, so the first thing I always say is to think about what is the question asking me. 2 models for the mechanism of an enzyme action. I'm kind of going to estimate the marks that I think might be available here, okay? So I reckon there's going to be at least 2 marks for the 2 models and explaining them. And then outline how changes in temperature can affect those. That's the second part. Outline how changes in temperature can affect those mechanisms of enzyme action. And I reckon there's probably going to be about 4 marks for that, okay? So, if it's 6, we should try and stretch ourselves and try and say maybe, 3 marks for that and sort of 5 marks for that. That's what we always want to aim for higher than the number of marks that are actually on the paper here, 6. All right, so let's go. So I'm going to just bullet point here and then we'll look at the mark scheme. Okay, so first of all, we got lock and key. And then we want to explain that. And then we've got induced fit. And then we want to explain that. We want to use words like, complementary. Keywords are so important. Complementary, we want to say substrate. We want to say active site. Enzyme substrate, hang on a minute. Yeah, enzyme substrate complex, ESC, that sort of thing. Doesn't change shape. And then this is, the shape changes slightly. We want to use all those words again, but we want to explain that for induced fit, as the substrate goes into the active site, the enzyme sort of accepts it and subtly changes shape around it, and then it reacts and then it releases it. Okay, so there's a slight change of shape. So hopefully we can get 3 marks for fully explaining all of that. Now we're going to do this bit over here. How am I going to do this? I'm going to take this over here, okay? So there's loads of things we can say. First of all, low temperatures. Let's start with talking about low temperatures, okay? So low, KE, few collisions, few enzyme substrate complexes, low rate of reaction, and then increasing temp, more KE, more collisions, etc. Kind of repeat this. We could even, just, I don't know if it'll be on the mark scheme, but we can even talk about Q10, maybe. Q10 coefficient by increasing the temperature by 10 degrees C tends to double the rate of an enzyme reaction at lower temperatures. And then after that, we can sort of then talk about higher temperatures. We basically want to talk about denaturing, but we want to fully explain that. So we want to talk about bonds, hydrogen bonds, for example, hydrophilic, hydrophobic interactions, the fact that they're disrupted. We want to talk about the tertiary structure or quaternary structure. We want to talk about the active site, changing shape, and all those things, okay? So yeah, loads of things for us to talk about. Let's have a look at the mark scheme now and see if I would have got 6 marks on this question. Okay, so we'll talk about the kind of levels of response in a minute. But first, I think it's just good to look through your answer and look at the types of scientific point that you can make. Now, the mark scheme here says that you should use a green dot to indicate where scientific points are made about the 2 models of enzyme action, and use a highlight square to talk about temperature. So we're kind of, we're dividing up the different points. So in terms of enzyme action, there's so many points we could say. Look at this, 18 different things you could have said to get the 6 marks. I love these essay questions because it's really an opportunity to pick up loads of marks. You can just say what you know, really, as long as you answer the question. Okay, so enzyme substrate complex, enzyme product complex. mention those. Products leave the active site. You've got to be specific. Lock and key, induce fit. Okay, so yeah, there's talking about that. I think in the plan we would have got maybe 3 or 4, probably 3, 4, maybe even 5 marks there. Probably didn't say this enzyme. So yeah, I'd say we've got about 4 marks from the plan picked up there. Great. Potential marks. All right, effects of temperature. We talked about kinetic energy. We talked about more successful collisions, enzyme-substrate complexes. We talked about decrease in temperature, reducing kinetic energy, fewer successful collisions, fewer enzyme-substrate complexes. Great, we talked about all those things. Active sites. Actually, I didn't use this terminology, optimum temperature. I probably would have done if I'd written it out in full, but we didn't bullet point that. And we talked about the bonds and the tertiary structure, changing shape of active site, denaturing. Didn't talk about this reversible and irreversible either. Is there even more? No, that's it. Okay, so I think we definitely would have got. I definitely would have got 6 marks there. But when you're marking this, let's really check what level of response you gave. So I'm hoping that most people would have definitely got at least this, level 1 or 2. So describe some aspects of the mechanism of enzyme action and describes an effect of temperature. So, okay, so this for example, would be maybe you talked about lock and key, and then you talked about denaturing. But you didn't really talk about low temperatures because you just talked about an effect. That would be something like this. 3 or 4 marks. One or both mechanisms of enzyme action. So maybe you mentioned lock and key and induced fit, but you didn't really describe them in detail. So you just kind of got points 4 and 5, but not 1, 2, 3. And describe some ways in which temperature affects the reactant site. So maybe maybe, for example, you talked a little bit about, low temperatures and just said that it was therefore a low rate of reaction, and you talked a little bit about high temperatures and denaturing, but you didn't really talk about kinetic energy, for example. Could be a level 2, 3, or 4 mark answer. So for the level 5, 6 mark answer, what we're really going for here, you've got to talk about both mechanisms of enzyme action, lock and key and induced fit. Clearly describe those mechanisms, talking about enzyme substrate complexes, that sort of thing, and then you provided a detailed description of the ways in which both high and low temperature affects the reactants and active site. Okay, so I think so that's how you answer that question. Just to recap, when we looked at the question and sort of planned it, unfortunately my notes have disappeared here, but what we did when we planned was we first just kind of highlighted the 2 different parts of the question that we saw, and we sort of said, okay, I wanted to get at least 2 marks about this enzyme action stuff, and then I want to get my 4 marks from the temperature stuff. So I had a strategy. Okay, let's move on to question 5. That was a big question, that question 4. So 2 more questions to go, 5 and 6. All right, on the insert shows the circulatory systems of 3 groups of animals. What type of circulatory system is shown in all these animals, and how does the circulatory system of a fish compare to a mammal? Here's the insert. Okay, so all 3 of these systems are closed systems, okay? In contrast to open systems. Remember, closed systems have blood vessels, open don't. So open would be an insect system, for example. And then the second part is, how does the circulation of a fish compare to that of a mammal? Okay, well, here's the fish, and here is the mammal. So a fish is a single system, isn't it? Okay, it is a single system, and this is a double circulatory system. Don't just say single versus double, that's not enough. Elaborate further. So the fish has a single circulatory system in which blood passes through the heart once per per trip around the body, whereas in the mammal there's a double circulatory system and the blood flows through the heart twice, and there is a separate, you could say, pulmonary circulation, and this would be called the systemic circulation here. Okay, it's not labeled. This pulmonary is labeled here. So if you wanted to use this terminology. So, for example, in the fish, the blood goes through the gill, capillaries of the gill, and then travels via the aorta to the capillaries of the body. Remember, we also said that in our mind map when we looked at this, we said that this is, this means that there's quite low blood pressure by the time it's getting into the body, whereas in the mammal system, it's more efficient because we can have a relatively low blood pressure in the lungs, powered by the right side of the ventricle here, the right ventricle, and then the left ventricle pumps at higher pressure, so we can have high blood pressure traveling to the capillaries of the body, which is a more efficient system. Amphibians are weird. I'm going to come back to that on the next question. All right, so let's look at the Marc scheme for those two things first of all. Okay, so fish is closed. The fish has a single circulation and the mammal has a double. Got to say both. You can't just say fish has a closed system. Okay, right. Another big 6 marker. Fantastic. Let's strategize again, okay? On the insert shows the flow of blood through the heart of an amphibian, such as a frog. Use the information. Yeah, let's definitely use the information. If it says to use it, we better use it. In figure 5.1 and 5.2 to compare the circulation of a frog and a mammal. All right, okay. So frog, mammal. Okay, well, let's look at similarities first, okay? So we've already talked about them, but let's just state them again. Just make sure we get marks, okay? They're both closed. They both have arteries, veins, capillaries. They both have a heart. Yeah, what else? Let's go back to this. Capillaries, arteries, veins, capillaries, they both have a heart. They both have a separate pulmonary circulation, I suppose. Yeah, they both have a separate pulmonary circulation, you could say. Those are the main similarities. What about differences? Okay, well, the clear difference is here, okay? The difference here is that there is only one ventricle, whereas in the mammalian heart, there are 2 ventricles. So that's actually more stuff that we could say in terms of similarities. So the heart has 2 atriums in both. Right, now let's look at the differences. So 1 ventricle, 2 ventricles. Big difference there. So we have oxygenated plus deoxygenated. I'm writing in shorthand here. Oxygenated plus deoxygenated blood mix in the ventricle, which is weird. And here we have a separation of oxygenated plus deoxygenated blood. So that's really comparing the circulations. Okay, so that's the first part. Okay, and I think that's probably worth about, probably maybe about 4 marks, what we said. So I still need to pick up 2 marks on this relative effectiveness. Okay, so effectiveness. Okay, so frog is going to be less effective. The amphibian, sorry. So, several reasons. Let's look back at the insert. So, well, the first thing is that this blood flowing down here, down the aorta to the body, is only mixed. It's mixed blood, okay? It's partially oxygenated. So, it's not carrying as much oxygen to the tissues as the blood which flows to the body tissues in the mammal. Here we have oxygenated blood carrying the maximum amount of oxygen to the body tissues, whereas in the frog there's less oxygen. Okay, so that's 1 point, the amount of oxygen that's carried. The second thing is that because the left ventricle, that's this ventricle here, left ventricle of the mammal, can be specifically adapted to pump at high pressure, then we can get the blood to the body tissues faster and at higher pressure. Whereas in the frog, if we had very high pressure from this ventricle, I don't know what it's called, if it's right or left, it's just the ventricle. If it was too high a pressure here, then the high pressure, if it was flowing through the pulmonary circulation, it could cause perhaps pulmonary edema, which is fluid leakage. So therefore, it won't be able to pump as high pressure. Okay, so those are the things we could say. Going back to the depth here, so frog less effective. So we need to talk about, just very briefly, we need to talk about the mixed blood, less O2, and then we need to talk about the ventricles, lower pressure pumping, which again is less effective. Okay, so yeah, 2 marks on effectiveness, 4 marks on the structure. Okay, that's my strategy. That's the bullet point plan. Let's have a look at the Marc scheme and let's mark your answers. Okay, again here in the marker's guidance, it says, you know, and this you can do this as well when you're marking. Green dot if you've made a point about the 2 circulations, and a highlight square to talk about the effectiveness. Okay, so that is, you know, those are the 2 aspects of this 6 marker. Right, let's look at the points. Okay, yeah, so we do get some of the points for some of the things that we've said. Did I say double circulation? I don't think I did I? So I could miss that point. Oops. Blood from mammalian heart transported separately to lungs and body. Did I really talk about that? I did talk about that. Blood and oxygen never mix. Blood from heart transported to lungs and body together. I think I talked about that. Blood going to the body in the frog is partially oxygenated. I talked about that. Oxygenated blood only separate when returning from lungs. I didn't say that. I didn't mention that. Reference to the spiral valve. Didn't talk about that. Where's the spiral valve? Where's that? Oh, duh. Hello. I didn't look at this part. There's a massive error, isn't it? I didn't look at this whole thing. So the spiral valve partially separates oxygenated and deoxygenated blood. Interesting. Okay. Didn't notice that at all because I didn't scroll down. Flow of blood through the heart described. I think I did do that. Reference to differences between structure of frog and mammalian hearts. Yeah, I did do that because remember I talked about they both have 2 atriums, but the frog has 1 ventricle, whereas the mammalian has 2 atriums and 2 ventricles. Effectiveness of circulation. Frog could be considered to be less effective. Yes, we've said that. Frog has less available oxygen for body cells. Yes, I've said that. Circulation is effective enough for the frog's needs. Didn't talk about this. Interesting. So basically, frogs have a lower metabolic rate than mammals because they are ectothermic. And this is interesting. So frog heartbeats beat faster to compensate. Didn't talk about this, although I did in the mind map actually. Frog oxygenates blood at skin and mouth. I think I read somewhere that it's around about 10% of the oxygen in an amphibian comes from their skin. Okay, mammalian, we talked about this. Body cells get maximum available O2, has a higher metabolic rate. Doesn't really talk about pressure actually, so I kind of, maybe I didn't get that mark. Now, I think I got 1, 2, 3, 4, 5, 6, 7, 8. I got 8 marks, maybe 8 things that I've said there. I wonder if I would have got 4 marks for that or not. I'm not sure if I would. Okay, so in terms of the levels of response, now time to work out what kind of level of response you got. I'm going to assume that we all got this. You either described the mammal or the frog. Okay, so you just did one of them. Okay, or hopefully you did describe both, and you commented on the effectiveness. So you described the mammal circulation, and then you said that it's a bit more effective. Okay, level 2. Again, describes the mammalian or the frog, attempts a description of, okay, so you described one well and you described the other less well, so maybe you described the mammal circulation quite well, but you didn't describe the other that well, and commented on the effectiveness of the 2 systems. For level 5, 6, you need to describe both the frog and mammalian circulations, give some detail of the relative effectiveness of the 2 systems. So I think I would probably give myself in that plan, I would say 5 out of 6, because I didn't really mention the spiral valve, and I don't think I really described the journey of the blood round the circulation enough in the frog, really. Okay, but again, just as a kind of an idea for approaching these questions, always think about the the Marc breakdown. 4 from the circulations and 2 about the effectiveness was my plan. All right, let's move on. Question 6, the last question. Okay, fantastic. All right, here's an experiment that was carried out to investigate the resistance of species of bacterium to the antibiotic penicillin. Okay, so we've got the bacterial colonies growing on the agar. So then we had this transfer. We will learn actually more about this in year 13. It's called replica plating. Basically, imagine you've got the agar plate here. You take a sterile cloth. It's actually sterile velvet, if you want to know, and you touch that cloth to the surface of the agar. It picks up some of the bacteria, carrying a print of it on the cloth, and then you go over here and you press it down onto a new piece of agar. So it transfers the bacteria in the same sort of orientation that they were on the first plate to the second plate. It's a clever technique. Only 2 colonies survived and continued to grow on the agar that contained penicillin. The bacteria in these colonies possessed a mutation that gave them resistance to penicillin. Now we take the original plate and flooded it with penicillin, and it's the same 2 colonies that are growing. That looks to me to be this one and this one, by the way. So these two colonies are the ones that had the mutation. All right, student made the following suggestions. I think that the colonies on the agar containing penicillin that survived and grew so did so because those bacteria evolved resistance. They evolved resistance as a result of being exposed to penicillin. I have a problem with this. This is sort of incorrect. See if you can think why. Let's look at the other student's comment. But some of the bacteria in the population were already resistant, so they can't have evolved resistance because they were exposed to penicillin. Yeah, that student's right. Well, the evidence is that as soon as they were transferred onto the penicillin, they could survive. It would maybe take some time, and furthermore, probably the better evidence is that when the original plate was flooded with penicillin, those 2 colonies survived. Okay, so there was already a mutation present that gave them resistance. It wasn't the exposure on the second agar plate, this middle one here. It wasn't this exposure that made them be resistant. There was already a mutation there. Name the process that increases the proportion of penicillin-resistant bacteria in the population. That is natural selection, okay? Ooh, what's going on here? Let's look at the Marc scheme. They're just checking that. The third diagram shows that resistant bacterial colonies are already present. These resistant bacteria on the original plate continue to grow when flooded with penicillin. That's right, natural selection. Great. Okay. Right, let's go to the last bit. All right, okay. One role for the Office of National Statistics is to collate data about the causes of death in England and Wales. Staphylococcus aureus, let's get the highlighter out, can be mentioned. Staphylococcus aureus can be mentioned on a death certificate and so may also be specified as being methicillin-resistant MRSA. Number of death certificates mentioning Staphylococcus aureus, 1993 to 2012. Okay, there's a whole lot of data there. I'm just kind of gonna, just scan it first before I look at the question. So it's sort of climbing, it's kind of fluctuating in the 400s. It seems to go down quite a lot here from 2010 to 2011. Let's look about this. Specified as MRSA, it's climbing, going up, it's going, oh gosh, it's getting super high. Super high. This is the maximum here, and then it's starting to decrease, a big decrease here from 12 to 7, and then it continues to decrease after there. And total, again, it's climbing, it's climbing, it's climbing. We've got a maximum in this year, and then it's decreasing. Okay, all right. Calculate the percentage increase in the number of death certificates that mention MRSA from 1993 to the year when the numbers reach a peak. Number of deaths. Okay, so this is the number of death certificates that mention MRSA. Okay, from 93 to the years when number reaches a peak. So let's find those numbers. So 93 is 51, and the year when they reach a peak is 1,652. So what is the percentage increase from 51 to 1,652? 51 to 1,652. Okay, so the percentage change is always equal to the change divided by the original figure, or the starting figure, times 100 to make it a percent. So the change is, calculator out, 1652 minus 51 equals 1,601, divided by the original, which is 51, and then we times it by 100 to make it percent. So I'm going to do that. 1,601 divided by 51 equals, times 100 equals, ooh, that's big. It says 3 significant figures there. So I'm going to write this. Well, first I'll write right over here. 3139.2. Now I only want that to 3 significant figures, so it's 3140. Is that the right answer? Yes, 3140. Great. The proportion of death certificates that mention MRSA in 1993 is 12%. Compare this figure with the proportion of death certificates that mention MRSA in 2012. Okay, so that's what I want to look at. In 2012, what have we got going? Okay, so there's total of death certificates is 507, and 292 mention them. Okay, so what proportion is that? 292 divided by 557 equals 0.52, or 52.4%. So this is a lot higher. And actually, I'm just going to do an extra little maths here. I'm going to divide that by the 12%, see how many times higher. So yeah, 52.4 divided by 12-ish is, it's over 4 times higher. Yeah. So let's see what the Marc Scheme says. I'm not sure if I got 2 marks for that. Was lower in 1993, has increased in 2012. So yeah, except, for example, over 4 times greater in 2012, and it's 52.4% or 52%. Yeah, that's what I got. Great. What can you conclude from these data about the deaths involving Staphylococcus aureus and MRSA since 2007? Well, since 2007, this has been dropping. This has been dropping even more, I would say, and this has been dropping. Because, okay, so this has gone from 1,600 to sort of close to 300. So that is a big drop. This has gone from, sorry, that's, yeah, close to 1,600 to about 300. That's gone from about 460. So that's about halved, roughly-ish. And that has gone down by a factor of 5 times or something. Yeah, that's about 5. Yeah, gone down about 5 times, and that's gone down about, reduced to about a quarter of its original value. So, all right. So the 2 things I would say. Deaths involving SA plus MRSA, both decreased. MRSA deaths have decreased more significantly. And then I would probably also put some figures in there as well. So I'll just do some calculations and say they've decreased by close to 3 times or close to 5 times or whatever like that. Let's look at the Marc Scheme. Decrease in number of deaths since 2007, yeah. consistent, steady, large decrease from the MRSA, so that's a bigger decrease. I didn't talk about this at all. I think I would have got 2 marks for that. Reference to better specific cross-infection control measures. Okay, but yeah, this is what I suggested you do. Any correct processed data. So talking about, what percent decrease it is, or approximately. I think I we had gone to about a quarter, so that's approximately 75%. Yeah, so kind of using some estimated figures. Okay. That is the end of the question paper. I hope you've found that useful. If you need to go over any questions again, you can always jump back to the part of the video that was about that specific question. I was really, really encouraged by your Marc's last lesson. So please can you scan this paper in using an app such as Cam Scanner and upload it to Teams, so I can have a look at your individual answers and see how you've marked your work and where you've done well, and also to sort of check your marking, because sometimes students can be a little bit too lenient, especially when it comes to those underlined words in the mark scheme. And thanks very much, and I'll set you another past paper next week, so I'll see you then. Thanks, bye.